So we've been working for the last few years in trying to build more physiologically complex human models of the bone marrow. The idea being that there's a need for human systems that capture the complexity of the whole organ to enable more translational research. And that's been our focus now for 5 or 6 years, which makes me feel a bit old. But yeah, essentially, we started off working in the context of myeloid cancers, and increasingly, as we build more niche complexity, the next big question for us was whether we can use this to study myeloma, which is a disease which has fairly critical elements of the microenvironment involved in its progression, and is a very real need for more effective treatments. And we think that human preclinical models are a way forward to those treatments and to meet that need. So I think the major challenge is whether we can maintain patient samples for long enough to be able to look at acquired resistance to therapies and clonal evolution and then mutations. And I think that's a major need not only in myeloma, but also across other blood cancer types. And hopefully that's something we can address in the coming years.